All right. Thank you, Jose, and I hope you still have some energy left. I'll try to go fast and show some movies and make it entertaining. So I'm new, so this area is new for me, and I learned a lot. It's one of the most fun and informative conferences for me. And actually what I learned today is that, as people were saying from the morning, that it seems like what we need is systems approach, and there are people here who have done that, but we need this approach where maybe you need thousands of things working together in terms of simulations to be able to understand what's going on. So quickly, these are the people that my former graduate students who did the work that I'm going to talk about. And Arpita Upadhyaya, I don't know why it gets cut at the bottom of the slide, well, is a long-time collaborator at the University of Maryland. And also I learned I have to do disclosures. So this is, I'm involved in computational drug discovery projects in 3 companies, as you can see. All right. So I'll start actually with a landmark paper, I think, from Marcus Covert's lab at Stanford. This was published in 2012 in Cell, which was one of the first whole cell simulations. And their lab was doing both experiments and simulations, and not abstract simulations, but trying to get, these are predictive simulations, meaning that they, on a tiny organism, which is, I think it's on a microplasmic bacterium, which is like almost like a nanobacterium with very small set of genes and so on, they were able to quantitatively follow various genes and metabolites and actually come up with a predictive model that they could verify. So it started open the field. And this slide, this actually is a paper that came out a few months ago from Zhen Schulten's lab at University of Illinois Urbana-Champaign, which in a way is a follow-up of that previous paper, meaning that it took sort of, I think it took those kind of models from Marcus Covert's lab and then put it in 3D. So using GPUs and 3D now stochastic diffusion simulation, you can see sort of this Minecraft-like way of, again, simulating this tiny bacterium. And again, it's a huge amount of work to put together all these networks. You can see here that we are talking about thousands of interactions and signaling and metabolites and so on. So 10, 20 years ago, this would have been mind-boggling, but nowadays it seems that maybe these things are possible. What both simulations, so Covert's simulation was 0-dimensional, meaning I think there was no space. This simulation is 3-dimensional, but things are not moving, at least largely. So it's like the membrane is not moving. It's sort of frozen in space. What inspires me is things like this. So this is from Eric Betzik's lab, where you see a T-cell activation, and you see cells interacting, and clearly they are moving, shapes are changing, and also things inside cells are moving. So I'd like to simulate that from fundamental microscopic principles. And also coming to neuroscience, we can, for example, look at the exon growth cones, and you can see again that there are these flows in the cytoplasm. Clearly, that must be important somehow. And it gets cut off here, but it says, what I like to do is, if you take signaling and metabolic pathways, that already was in these previous 2 slides, and add gene networks, which was also in these previous 2 slides, but now add 3D mechanobiology, that's what I'd like to bring in, then maybe we can start to simulate these things. And I think in terms of neurodegeneration is that if we had models like were in this Marcus Covert's model, where you have thousands of genes and signaling and metabolic networks, maybe that will give you to this holistic thing where you don't have to worry about single pathway or single protein, single target, but you will have thousands of pathways interacting and giving you phenotypes, and you can then run these simulations in a computer. So our efforts were culminated in this set of algorithms that we wrote. It's called Mechanochemical Dynamics of Active Networks. We call it MEDIAN. And as you can see, there's a website, so you can download. There's tutorials. We actually have trained graduate students from other universities to use it. But the main idea is that to do the part that you already saw in Zhen Schulten's work, which is how about we compartmentalize space into voxels of some sort, and we can have reaction diffusion, or you can have thousands of reactions and molecules can diffuse around. We can have both passive and active transport. There are molecular motors that can walk on filaments and transport vesicles. We can do that. Our work last 6-7 years have been largely focused on cytoskeleton, but we can do pretty arbitrary chemistry. But in the cytoskeletal area, we can do polymerization, depolymerization, capping, and I think I just have a visual version of that. So if you have a cytoskeleton like an actin network, those networks can, for example, if there's an end of an actin network and there is a G-actin molecule that's diffusing in space, it could polymerize or it could depolymerize. Cross-linkers can bind and unbind. Molecular motors can bind and start walking and shearing the network. And because it's shearing the network, then you have to do forces. So we do have a full-blown mechanical model behind this too. And then on top of that, we also recently implemented a membrane model. So we triangulated a membrane. And our triangulation size, I think for you to give some scale, is that it's about 20 to 30 nanometers. Think of a triangle with the side of a 20 nanometer. So it's not at the level, so this is coarse grain. This is not at the level of a single lipid or even a few lipids. We're talking about maybe thousands of lipids in a single triangle. And that triangle, you can see it here. It's small compared. Cell scale is large compared to molecular scale. So this is whole another new scale, new physics in some way that we have to think about and use and treat. And then with this membrane model, we can include the surface tension of the membrane and its bending and osmotic pressure, things like that. Recently, we also have implemented reaction diffusion separately on the membrane. So you have, for example, receptor proteins that diffuse around, they aggregate, they signal. We can make receptor proteins curvature dependent. So for example, they could induce protrusions, things like that. And it all then culminates in a cover. And then I want to show you a movie. Yeah. Let me go back. Sorry. I have this lag time. OK, let me try again. So we can do a simulation where inside there is this 3D reaction diffusion. And this thing actually is smoother on my computer than for some reason it's on Zoom. But you can see that we can get membrane deformation. If we want, we can get reaction diffusion of, for example, surface receptor proteins. We can. And my iPhone, my Apple Watch is talking to me too. Anyway, we can sort of have even, in principle, we can have external signaling that through the membrane could induce then surface receptor protein aggregation, and that can induce downstream interactions. This is and I'll give you a couple of examples of using this. So the first example is a collaboration with Arpita Upadhyaya, who is at the University of Maryland. And she's a biophysicist who is working on T-cell activation. She puts them on gels and looking at things like forced traction, but also, in particular here, she was looking at actin and myosin. And often in T-cell activation, you see these rings forming at the periphery. And they first spread and they contract, and clearly it's sort of related to the phase of the activation. And why exactly they form? Why do they? You can see that the green and the purple have this two-ring structure. All these things are not really known. So we found that our simulations actually could reproduce those rings in very basic ways. And not only that, but we could have pharmacological interventions. For example, we could accelerate actin treadmilling, or we could inhibit myosin, and we could reproduce what's in the experiment. So here you can see, for example, that by inhibiting myosin or by accelerating myosin, the cell systems, you sort of, in one case, you can see that the ring constricts. In another case, actually gets disordered. And then we can do simulations, and we can reproduce many of these phenotypes. An ongoing project, long time, but still we haven't it's still sort of an ongoing project for us, is to do molecular model of a dendritic spine where we include multiple proteins and cytoskeletal components and signaling molecules that define the spine. The final project that I'll tell you about that we have done, there are two papers that are under review on this, is that we collaborated with Ed Ginniger, who is at NIH, working on exon guidance. So they do live embryo microscopy, where they see exon PET finding, that there are these exons that they grow and then they turn. And what they do, they actually follow Actin Dynamics. So all the colors are a little bit off here. but in an exon, and I can see a movie here, that they look at a growing axon, and the axon makes these protrusions and it makes turns. They also at the same time can monitor actin dynamics, and they see that actin dynamics to a large degree is important in how in axon protrusion, and in particular they are trying to understand in terms of signaling what controls those protrusions, and here I'm giving you at the bottom you can see that we're going to think about scales from nanometers to microns to hundreds of microns, and in terms of time scales, microseconds, milliseconds, minutes, and hours. And start from here is that, so they're interested in this Abel kinase, that they modify Abel kinase, they do knockouts, upregulate, downregulate, and then they get phenotypes that in some sense are biological phenotypes, let's say at a multi-hour time scale. And then if you do sort of this quicker experiments, then you can sort of connect this to a certain degree to actin dynamics. So you can see that there are actin phenotypes. And on the other hand, you can go all the way to signaling pathways. And this Abel, on one hand, it produces this linear filament growth, and also through this, it can also do branch filament growth. So somehow there's this multi-scaling or emergence, if you'd like, that somehow this signaling network locally ends up modifying actin dynamics at those larger timescales, and then later on that ends up having an effect on the biological phenotype. So that's what these kind of simulations, or both experiments and simulations, try to understand, like how do we go from molecular processes to these larger biological phenotypes. In this work, we were actually only able to, at least on the computational side, to look at this part. And that's also in these large-scale simulations of thousands of genes and metabolites, this is our situation, that we don't know most of kinetics there. So at best, what we can do, we qualitatively know that ARP23, so there are these two proteins that this Abel protein influences ARP23, which is a dendritic a protein that induces dendritic growth, versus which are linear filaments. So basically, we are going to look at various combinations of ARP23-driven growth versus INA-driven growth. And this is from Nash's movie, and I think that's how Arvind felt when he was doing this project, sort of looking at the formulas on the glass. These are his slides, so so basically, then we can, so he has done a huge number of simulations, and these tubes that we do, that you can see here, they are about 7 microns in length, and they are 1 to 2 microns in diameter. So for those of us that are doing molecular simulations, this is huge, right? So, and we can, the vertical axis is ARP23, horizontal axis is INA, and in particular, you can see that the primary determinant of what's going on is actually ARP23, that by varying ARP23 concentration, this network can become widely distributed through the whole thing, or be contracted like here. And also, when we look at the distribution of actin, we have distributions that are pretty flat versus distributions that are peaked. And we match experiments with these relatively low ARP23 concentrations. And in particular, the main thing that we basically see is that either we get this homogeneous actin in the tube, or it can get fragmented. And in particular, let me go. So we have this fragmentation process, and actually there's this coarsening process that various fragments exchange material with each other. So there's a quick formation of these clusters, and then they start to, on a slow time scale, exchange particles and coarsen. And this is the last slide I'll show you. It's a movie that he came up with a clever, so the whole project that then came to this idea of fragmentation, that under certain conditions, the actin network can fragment and lose coherence. And in particular, it would lose mechanical coherence. Meaning, if a network is mechanically coherent, if you ping it on this side, you will feel on the other side. If it's not coherent, it's disconnected in a way, then you won't. So that's what he has done with these simulations. You'll see that he is basically pulling them. He is like putting this thing and pulling the network. And in this case, he can pull the whole network with it. But at the bottom, you can see that this is pulling only part of the network. So this shows that the network gets mechanically fragmented as you increase the up-to-tree concentration. And these things actually, I just have, I think, one slide, slide at the end here. Let me just leave it here like this. So this thing actually shows that it is, first of all, closely correlates with what Ginniger's lab is observing experimentally. And in particular, they also see this actin network fragmentation. And the current work is to further sort of to add more details and be able to come up with these predictive models of exon PET finding. And my final thought on this is that, again, neurodegeneration is not something, I think this is not something that we've worked on, but I'd like to work on it using these kind of models. And I think basically what this brings on is this systems level approach, right? That we could have hundreds or maybe thousands or maybe tens of thousands of proteins and genes and metabolites interacting in 3D, where you have both structural changes at the level of the cell, geometric, mechanical, and see, can we come up with predictive models of some of these phenomena that we have discussed today. So, and that's all. Thank you.
So we have time for a couple of questions, or people can ask general questions to all the speakers.
Garik, to make this a little more concrete for me,
Okay.
Take the example of an actin monomer approaching the fiber. How does it decide whether to elongate or not?
So basically, you have an actin, let's say, network, and there are these ends, and then there's an actin monomer that's diffusing around. So if it's far away from any of the ends, it's not going to polymerize. But if it's, let's say, it's very close, then there's a probability that it might, right? We know from the rates and so on, we know what that probability is. So we have a master, in chemistry, we call it chemical master equation. So it basically probabilistically decides based on the known kinetic rates whether it should or not. So everything is stochastic.
Thank you.
So since you talk about stochastic, and you finish with this directional neuron, here you're just showing fragmentation. How are you going to direct this fragmentation?
So that's why I think it will take maybe a number of years to get this worked out. Is that there are these philopodia that so clearly somehow this fragmentation is having an effect on how philopodia get projected, and then sometimes the body of the actin mass then goes into one of the philopodia, and that becomes the new sort of axon. But these are kind of things that we try to work it up from step by step. So these are the first two papers. I think we'll need more work to answer your question.